Okay, hello and welcome to the ZOOMinar. Today's speakers are Dr. Avindra Nath from NIH and Dr. Bitan from UCLA. I also would like to welcome Dr. Danila Miladi from Catania, Dr. Raj Jelinek from Ben Gurion University, Israel, to the panel. Also, Dr. Varun Govind to the panel. He's from University of Miami. I'll pass the link to Dr. Govind. Varun Govind.
All right.
Go ahead and get started.
Yeah. Thank you, Dr. Rams. Good morning or good afternoon, depending on where you are in the world. So I'm happy to introduce Dr. Nath today. Dr. Nath received his MD degree from Christian Medical College, Ludhiana in the state of Punjab in India in 1981 and completed a residency in neurology from University of Texas Health Science Center in Houston, followed by a fellowship in multiple sclerosis and neurovirology at the same institution, and then a fellowship in neuro-AIDS at the Neurological Institute of at the National Institute of Neurological Disorders and Stroke, NINDS, which is an NIH institutions in the US. He held faculty positions at the University of Manitoba in Canada between 1990 and 97, and the University of Kentucky between 97 and 2002. In 2002, he joined Johns Hopkins University as professor of neurology and director of the Division of Neuroimmunology and Neurological Infections. Subsequently, he joined in NIH in 2011 as the clinical director of NINDS and the director of the Translational Neuroscience Center and chief of the section of infections of the nervous system. His research focuses on understanding the pathophysiology of retroviral infections of the nervous system and the development of new diagnostic and therapeutic approaches for these diseases. Today, Dr. Nath will talk to us on the topic of aggregates of HIV-Tat protein complexed with amyloid beta peptide accelerate neurodegeneration. With this brief introduction, I welcome Dr. Nath to this presentation. Dr. Nath, everything is yours now.
Well, thank you very much for that very kind introduction. I have to say I'm really delighted to be invited to this lecture series that you have. And the last 2 years, all I've been doing is talking about COVID. So it's such a relief to be able to talk about stuff that we've been doing for so many years. And this TAT aggregation is really a core of a lot of work that we've done over many, many years. And I'm absolutely, it's been a real pleasure to have the opportunity to really talk about it to individuals who really care about protein aggregation. And so, as you mentioned, I'm at the National Institutes of Health here, which is a suburb of Washington, DC. And the building that you see here is Building 10. And so these two buildings together form the clinical center. And that's where it's the largest brick building in the world, they say. And it's also the largest research hospital here. So we bring in a lot of patients on various clinical protocols. And my lab is right within the clinical center. And so it makes it very easy to see patients and do research with patient-related material at the same time. Okay, so having said that, one of the major areas of research that I've been involved in since the beginning of the AIDS pandemic is HIV infection and its effects on the brain. And HIV will cause a dementing illness if it's untreated. And it can cause cognitive abnormalities, motor dysfunction, and behavioral abnormalities. And if you look at this MRI scan of an individual who had HIV infection, this is a 30-year-old individual, and you can see that there's a lot of, black space over here, suggesting that there's a lot of atrophy of this brain, and mainly in the frontal lobes in the front of the brain, while the back of the brain or the posterior parts are relatively spared. So the question really is, what is causing all this destruction of brain tissue? Now, we think that with all the antiretroviral drugs, we should be able to impact it. And that is true. It has changed the way people live with HIV infection, and the mortality rates have gone down drastically. However, the cognitive abnormalities still persist. So although people don't develop this rapid dementia, but they do continue to have cognitive difficulties. So here in this chart, you can see in the pre-antiretroviral era, and then the black bars are in the retroviral era. And you can see that learning abilities are actually still even greater now, and executive dysfunction is also greater in these individuals. So there are certain parts of the brain that are not really responding very much to antiretroviral drugs, and I'll explain to you why. And also, if individuals have cognitive abnormalities, it has impact on survival, because if you can't remember properly, you won't remember to take your pills, you may pose yourself at increased risk of various kinds of things or risky behavior. And so, as you can see, that even with antiretroviral drugs, those individuals who develop cognitive abnormalities, their survival is much, much lower. So what happens to the virus? You might think that, okay, if there's that much neurodegeneration taking place, you're going to find the virus in the neurons, but that's not the case. You do find virus in the brain, but it's infecting the macrophages of microglial cells and astrocytes. So it comes from the blood, and it enters through these monocytes, either as free virus or on these monocytes, and can infect these macrophages and astrocytes. The life cycle of the virus in these two cells is quite different. So if you look at the perivascular regions, just around the blood vessels, you find all these infected cells here. And this one is immunostaining for the TAT protein here. You also find these multinucleated giant cells. These macrophages can coalesce with one another and form these multinucleated giant cells. Okay. And here it's immunostaining for the envelope protein. So it's a productive infection in these macrophages. Now here I want to show you, this is a microglial nodule, and these small brown dots here, even though at low magnification, they're sitting as small aggregates within the microglia and microglial nodules. Okay. The second cell type it infects are astrocytes. Now the astrocytes become latently infected. So you can see some viral products there, but you don't see all of them. So that suggests that the virus becomes a reservoir in these astrocytes, and it can produce some viral proteins and release them extracellularly, but the cells themselves never degenerate. Okay. And that's important because what I'm going to show you is that through these cell types, TAT protein can be released extracellularly and can cause aggregation and toxicity. And this is the map of the viral genome, and the proteins on top are the first ones to be formed. And so the TAT protein is right on top, and that's the first protein to be formed. Now, looking at neurons, if you look at the neurons, here is the dentate gyrus, and this is nicely staining neurons here all along the dentate gyrus. This is an HIV infection with no encephalitis, HIV infection with encephalitis, and then with drug abuse. You can see that the amount of staining decreases, suggesting that there is neuronal loss as you go forward. Okay. And here at high magnification, again, this is what a normal brain looks like. And this is an HIV demented individual. And you can see that there's paucity of staining. However, the interesting thing is the neurons are dying, but there's no infection of the neurons. So the death of the neurons is all indirectly mediated. Okay. So what happens is that HIV infects these macrophages and the astrocytes, and then they release these viral proteins that are then toxic to neurons. And so we found that even in individuals who are on antiretroviral therapy for extended periods of time, here over 10 to 15 years of a good control of the virus, you can still measure TAT protein in their spinal fluid. So these are, each one of us is a separate individual here, and the amount of TAT was measured here and on the y-axis. And you can see that about a third of these individuals have TAT present here. And you can measure it freely in the CSF, and you can also see it in exosomes. Okay. And those individuals who have TAT that is detectable, they also have cognitive impairment here. So it's shown as the psychomotor domain and information processing speed is decreased. And by immunostaining also, we can show in individuals, and this is a biopsy specimen, and we can see TAT staining macrophages that are present in the brain, despite the fact that virus is well controlled and you cannot detect P24 here in these individuals. So now here, oops. So here you can see in an individual here that there are all these TAT aggregates that are present in this inflammatory milieu here. And so when the protein comes out, it self-aggregates. Okay. And I showed you this one already. This is so if you just take the protein and you put it on neurons, it will, interestingly, it'll bind to neurons and it will cause toxicity. So the monomeric form or the dimeric form are two that can cause neural excitation very, very easily. And so each bar here is an action potential. So that means as soon as it touches the neuron, it just starts firing like crazy. Okay. And because it aggregates, it can sit on the neural cell membrane and just continually keep exciting it till the neuron finally dies. It can also act on glial cells, causing activation of the glial cells. So here you can see release of IL-1 beta and IL-6 in a dose-responsive manner. So it doesn't kill the glial cells, it activates them, but it will kill the neurons if you give enough of it. We made a transgenic mouse model with the TAT protein. And again, what we found was that, and this is an inducible animal, so if you give a doxycycline, then TAT gets produced. And what we found was that you can see that there's toxicity. So these are caspase positive neurons here. And you can see that there's increased number of caspase positive cells showing neuronal cell death once you induce TAT production in these transgenic animals. And again, shown by immunostaining and and you can see that in these animals, again, you can find the monomeric form, dimeric form, and tetramer. And there are also multiple polymeric forms of TAT that are present here at higher molecular masses. So TAT does oligomerize in these animals where it's being produced. Now, all TATs are not the same. HIV does mutate. Conserved regions are absolutely important. And so you see pretty much the same pattern. There is something called clade B and clade C. So they're a bit divergent from one another. And one of the differences is this cysteine at position 31. So that gets substituted by serine. And when the substitution takes place, its ability to cause toxicity is actually decreased. Okay. So this is one critical mutation that can affect its aggregation as well as toxicity. So in these transgenic animals, when we induced TAT, one of the other interesting pathologies that we saw was aggregation of amyloid precursor protein. So we found increased expression of amyloid precursor protein. So the question was, how does it actually do that? So I'm going to go through this in great detail, but this is what we think TAT is doing. What it does is it activates beta secretase. And by activating beta secretase, it's going to form a lot more amyloid beta peptide from the precursor protein because that cleaves it in order to form this. The second thing what it does is it blocks the uptake of amyloid beta into neurons. And by doing so, there'll be accumulation in the extracellular space. And lastly, what TAT does is it also blocks its degradation by interacting directly with neprolysine and inhibiting it. So at multiple different levels, TAT can lead to increased production of amyloid beta peptides. And lastly, I will show you that TAT and amyloid beta peptides can directly interact in order to cause aggregation and toxicity. So here's the interaction with base 1. Okay. So here, what happens is if you look at this on the right-hand side here, with exposure to TAT, you can see that the base activity actually goes up. Okay. And not only that, the total amount of base being produced also goes up. So you can see here in the Western blot here that there's more base protein and it is also more active. So when it does so, then it's going to produce more amyloid beta peptide. The second thing what TAT does is it acts on this LDL receptor. And by doing so, it blocks it. And by blocking it, there are a number of things that are taken up by neurons via the LDL receptor. And one of them is the amyloid beta peptide. And so, and I've drawn this little line around this to show you that this last bar here is TAT protein. And in the presence of TAT, amyloid beta peptide is not taken up, okay? And this is a positive control, and these two are negative controls here. So clearly, presence of TAT will block the uptake of amyloid beta peptide. Okay, then, so I showed you that that's a possibility. Now, the next thing is its interactions with neprolysine. So before I show you the exact interactions with neprolysine, let me just show you here in these immunostaining sections. This is the control. This is a HIV-infected brain tissue, and then patients with Alzheimer's disease, and the immunostaining for amyloid. Okay, so all this brown staining is amyloid. As you can see in the amyloid, in the Alzheimer's patients, you can see a lot of accumulation with amyloid, and at high power, you can see this really crisp immunostaining. In the HIV patients, you can also see accumulation of amyloid. However, the borders are a lot more diffuse. Okay, so these are called diffuse plaques. So you see a fair bit of amyloid accumulation. The difference is that you don't see a lot of tau accumulation like you do in patients with Alzheimer's. But the amyloid accumulation is quite striking. And here they quantified the amount of amyloid beta plaques that were found according to the duration of infection. And the longer the duration of the infection, the greater is the accumulation of amyloid beta amyloid plaques. So here then this group by Lynn Pulliam at UCSF, what they did was they looked at neprolysine activity, and they found that the amount of inhibition goes up in a dose responsive manner here with TAT protein. And then what they did was they made peptides, 15 more peptides overlapping with each other to try and see what really causes the inhibition of neprolysine. And they found that the cysteine-rich region is absolutely critical. So as I showed you previously, this dicysteine motif is in position 30 and 31. And one of these cysteines at position 31 can be mutated to a serine in clade C HIV-Tat. So that would be expected to cause less aggregation as opposed to the one that is more predominantly found in United States and North America. Okay, so now what I'm going to show you is that, yes, you can cause this accumulation of TAT amyloid, of amyloid beta peptide, but now the two proteins can also interact with one another directly, okay? And when they do so, they can cause more toxicity. So here what we did was we incubated amyloid beta peptide with TAT protein. And you can see here in this bar graph that with the increasing concentrations of TAT, while amyloid beta peptide is constant, what you can see is that, so it's a subthreshold concentration of A beta. And you can see that there's increased toxicity. So this is the cell puncta is a measure of neuronal toxicity, because the neuronal processes, once they start degenerating, they accumulate these little puncta in them. So you can count the number of puncta. And you can see here in the bottom that, with media, you can see this nice branching of these neurons. You put the amyloid beta peptide, this still looks like pretty nice branching here. And as you increase the total amount of TAT, you can see with 180 nanomolars, you just see these little dots all over the place. And these are these puncta that form on these axons. Okay. It's an indicator of neurotoxicity. Okay, so then Alina Hadigan was a postdoc in my lab, and she did some atomic force microscopy to try and understand how these aggregates are actually being formed. Okay. So this is just TAT protein by itself. So when you do AFM, what you find is that you find some monomers, and this is a monomer here at high magnification. And then she found some dimers over here. But what she also found was that there were a lot of multimers, and the multimers could be as large as 32 mers. There were some 14 mers. So certainly this is an in vitro situation where we put the recombinant TAT protein in our buffer and we're looking at it. But it aggregates very quickly and to form very large molecules. But the interesting thing is these aggregates are very spherical. And that is the least energy forming state for the TAT protein, is to form these spheres. If you look at amyloid fibrils, so you can see that amyloid will form these really nice uniform fibrils by themselves, okay? And they form these very nice uniform twists as well. However, if you incubate it along with the TAT protein, what will happen is that you'll see this decoration of TAT on top of these fibrils here. So it changes the conformation of these amyloid fibrils. And the distance between these twists will increase along as you increase the concentration of TAT. And it also makes it more rigid. This is, again, I showed you this already, but to show you that at these higher concentrations, you can see that TAT accumulates on these fibrils here, and it also fractures them, causing pretty large plaques of TAT and amyloid together. And so this is our hypothesis as to what is really happening. We think that the amyloid fibril is a uniform strand. TAT will sit on top of it. And by doing so, because TAT itself aggregates, so it'll bring all these other fibrils in proximity together, and it forms these twists. So you can form these very large twists. And then after a while, they become brittle, and then they'll start breaking apart. So as soon as they start getting fractured, now what happens is these smaller forms will again aggregate even further. And by doing so, you'll form these pretty large aggregates. Now, it is traditionally thought that, that amyloid plaques by themselves are not really toxic. What is toxic are the oligomers of amyloid. And that it's the body's defense mechanism to form large plaques and prevent them from causing neurodegeneration. What we showed you is that actually these larger plaques, when they bind with TAT, become extremely toxic. So the virus now takes what is body's defense mechanism and turns it against itself. And these, by charge-based mechanism, will form small pores in the cell membrane. And by forming pores in the cell membrane, will increase ionic exchange and leading to toxicity of these neurons. So in conclusion, I would like to say that TAT protein is released from HIV reservoirs despite adequate antiretroviral therapy. TAT can lead to accumulation of amyloid beta peptide by increasing its production, decreasing its uptake, and preventing its breakdown. TAT can self-aggregate and directly aggregate with amyloid beta peptide to cause neurotoxicity. So I'd like to stop here and acknowledge a lot of people who have done the work over the years, and particularly Alina Hadigan, who did all the atomic force microscopy work to directly show us how TAT was aggregating along with amyloid beta peptide. So I'll stop here. I think I kept you on time.
Yeah, thank you, Dr. Nath. So let's see the questions. Surprisingly, there are no questions I see. Any of the panelists, any questions to Dr. Nath?
Okay, I have just a curiosity. Avin, you just mentioned sometime that there is a critical mutation, serine cysteine, to activate neurotoxicity of the TAT sequence.
Yes.
But it is not clear to me if this mutation is related to the possibility to make intermolecular cross-linking by disulfide bridges, for instance. My question is, have you tried to reproduce these experiments in a reducing environment?
Yes. So TAT is a very basic compound, protein, and the PI is about 9.4. Okay. So that's how it really binds to membranes really well, because it's so positively charged and the membranes are negatively charged. So yes, it does matter. So we've looked at a lot of different conditions, and yes, you can change the aggregation properties by using reducing agents, but you have to use very high reducing agents. So even with SDS and beta mercaptan ethanol and all the kind of stuff, you can still have aggregates on your gel. So it's not easy to rip it apart once it forms these aggregates. Now, initially we thought maybe it had prion-like properties because we were making recombinant proteins and we couldn't really rip them on a gel. But with proteases, you can degrade it, so it's not like scrapey-like properties, but it's pretty close. You can form these very strong aggregates that are hard to rip apart.
Okay, thank you.
Good question. Very interesting talk. The curious observation for me was that you get a lot of amyloid pathology without tau pathology, which is not a common observation.
Yes.
We know that there is a temporary gap between amyloid toxicity and tau pathology. So I'm wondering if over time there is a correlation with some increased tau pathology.
Tau pathology, yeah. So it's quite possible. Now we have an aging population with HIV infection. And people who are infected in the 80s and 90s are now getting older. So as they come to autopsy, there's an opportunity to look at it to see if maybe tau is now a predominant part of the pathology or not. But yes, you're right. That question remains totally unanswered. Certainly, there's not a huge amount of accumulation of tau at an early age group. There are other diseases in which tau will accumulate early. With chronic traumatic encephalopathy, you can see tau accumulation. There's a condition called nodding syndrome that occurs in children in Uganda. There you can see tau accumulation. Patients with epilepsy, chronic epilepsy, you can see tau accumulation. So we're not seeing that kind of tau accumulation in this population. Yeah.
Right. The curious point for me is that you do see a beta toxicity without tau accumulation. That's the conundrum. So a couple of thoughts about that. One is maybe you need internalization of a beta in order for tau to detach from the microtubules and start to form aggregates. And the other one is there are some new papers suggesting that A beta oligomers facilitate tau spreading from cell to cell. And if you're sucking up all the A beta into those TAT associated fibrils and have no oligomers, that might explain why you don't see a lot of tau pathology.
Yeah, that's fascinating. Yeah, thanks very much for those comments. Yeah, I wasn't aware of it, but that makes a lot of sense. Certainly worth investigating in this context. Yeah. Yeah, very nice.
Oh, great talk, Avi. This is a nice work. You mentioned about the A beta fibers bound with TAT monomers, I guess, monomers or oligomers. They are super toxic. They form pores in the membrane. Is that right?
Yes, yes, that's
So
correct.
do you have any evidence for pore formation as opposed to protein interaction-based toxicity or other mechanisms?
Yeah, so it's indirect evidence. We have not directly visualized the pores. So in our Nature paper, we showed some indirect evidence and and then hypothesized the rest. But yes, I think it would be nice to be able to have some visual higher resolution microscopy to actually see the formation of these things. I think that would be nice.
So are these TAT oligomers or monomers?
Yeah, the oligomers, yeah.
So, Avi, I think in one of your slides, you showed neuronal loss in different HIV plus comorbid conditions, such as with encephalopathies with substance use disorders. Was that study done using human brain
human.
cell? Human.
Yeah, what I showed you was all human brain tissue. Yeah, that we did ourselves and on autopsy tissues. Yeah.
Autopsy tissues. Okay. Since you labeled that slide unpublished, so I thought, okay, let me check that. Okay. Are there any questions from anybody else?
There is a question in the Q&A and chat box.
Right. I see that. Dr. Nath, can you see that question or do you want me to read it? Okay, let me read that question for everyone. So this question is from Murugesan Velayudam. So the question is, does COVID-19 virus induce amyloid beta aggregation in the neurons?
Oh, COVID. Okay. So there's a lot of interest in it. So I've been on many panels. The thing is that we know that patients who have pre-Alzheimer-like conditions, they can develop increased cognitive abnormalities. And the concern is that maybe there is increased protein aggregation in patients with COVID, and that we may see not only more Alzheimer's, but Parkinson's disease because synuclein and amyloid and tau and all these things. So that jury still remains open as to whether that happens or not. Now one possibility is that it may not just be COVID. With any respiratory infection, when you get inflammation, you're going to get aggregation of proteins just through the inflammatory process. But is there something specific about COVID? We don't really know. But there's a huge amount of interest, and the National Institute of Aging has done a couple of interesting symposiums on this topic. And I think they're going to hopefully provide funding to study this further. So I think that would be good. I see another question here about the mechanism of TAT secretion. So if we know it's actively secreted, so it's an energy dependent process, but how it actually gets released has not been studied at all. I think people have studied uptake, and the uptake is through the endosomal pathway, but how it gets released actually is an interesting question. I think if somebody would look at it, that would be terrific.
So you primarily focused on TAT protein. So what about GP120? Is there any role of GP120 with amyloid beta, that's aggregation?
Yes, so there is one paper on it done by this guy, Norm Howie and Mark Mattson. They looked at GP120 and they did show increased, so it was, I think they crossed, did they cross two animals, or they injected GP120 into the, I think they injected GP120 into the amyloid precursor protein transgenic mice, and then showed plaque formation there. So that's the only paper that I know of. Yeah. But it wasn't pursued any further.
Maybe you mentioned about exosomes. I would assume exosomes
Yes.
are neuronal cells. These are exosomes containing A-beta aggregates or TAT aggregates. Can you give some more information on that?
Okay, so what we did was we pulled out the exosomes from the spinal fluid of these patients. So they were all types of exosomes. They were not further fractionated. So by differential centrifugation, we pulled them out. And there, our interest was really to see if we could find HIV products. So we analyzed them only for TAT, and we looked at TAR. I had it on the slide, but I didn't point out to it. So the TAR is the RNA domain that TAT binds to. And so we found both of them, although they weren't always concordant. In some exosomes, you could find TAT, and some you could find only TAR. In others, you could find both of them together. But we did not analyze it for other proteins. I think that is a good point, and we probably should go back and look at it, because we have a lot of spinal fluid we collect on our patients. I have a cohort of about 400 people that I follow here at NIH, and we've done spinal taps on a lot of them. So there is a good question. If anybody of you are interested in collaborating, we have lots of samples, actually. I'd be delighted to share them with you.
Thank you.
Alrighty, if there are no questions, let's take this opportunity to thank Dr. Nath for giving such a nice presentation. So Dr. Nath, as Dr. Rams indicated in the beginning, if you can stay with us until end of the second talk, that will be nice. If you can, stay. Otherwise, you please join us at about 1 p.m. to take additional questions, if any, at that time.
Okay, thank you very much. Yeah, I'll be here for that.
Thank you.
Ross, go ahead.
Yeah, thank you.
So I should introduce Gal now, yes?
Yes, yes.
Okay, so it's really my pleasure to introduce Gal, a good friend whom I known for too many years already, just tells us how old we are. We actually met and we spent the bachelor degree time in the Hebrew University in Jerusalem. Gal then stayed at the Hebrew University and got a PhD in organic chemistry. Then he moved to the Boston area and he spent several years at the Brigham Young Harvard Medical School, Brigham and Women's Hospital, where he did a postdoc. Since 2004, Gal moved to the West Coast and he's now a professor of neurology at the David Geffen School of Medicine at UCLA. Gal has made several very interesting and very important contributions to the amyloid field. One of the most notable, actually for me, is the discovery of small molecules that has exceptional capabilities of blocking oligomer formation of beta amyloid and other amyloid proteins. This was actually a basis of a company also and a business initiative, and lots of very exciting discoveries about the various amyloids and also antiviral properties, which is not what Gal is going to tell us about today, is what, from what I understand from the very interesting title of his talk. So without further ado, please, Gal, and the floor is yours.
Thank you very much. I'm going to share my screen. I assume you guys can see this.
Yes.
Okay, so I thought I'll take the opportunity today to discuss a new project in the lab, not the small molecules, but the discovery of new cellular structures called exophers in mammalian neurons. And this discovery follows some recent papers in C. elegans. Therefore, I'm showing you the evolutionary path from worms to humans. And I hope I can show you the similarities and the differences. So the story starts with proteostasis. We all know that cells have to make new molecules all the time, fresh molecules, and degrade the old ones. There are several degradation mechanisms, including degradation by specific enzymes, the ubiquitin proteasome system that degrades ubiquitinated proteins, mainly soluble proteins, and the autophagy lysosome pathway that degrades all sorts of molecules, including protein aggregates. So all of those mechanisms degrade proteins intracellularly. Every cell is responsible to taking care of its own trash. But a few years ago, the group of Monica Driscoll at Rutgers reported a new mechanism they found in C. elegans when they labeled specific light touch neurons in these worms with fluorescent proteins. And they observed that some of those neurons over time pushed a lot of the fluorescence into a new space. This is shown here in A, over time, developing this new thing that evaginates out of the membrane and eventually separates from the membrane, containing a lot of the fluorescence. This is the cell, the original cell. And they call these things exophers, meaning taking things out. They looked at the fluorescence and they showed that the nucleus obviously remains with the original cell. The fluorescence that gets pushed out in the exopher never shows nucleus or significant amount of DNA. They also showed that the exophers can be connected by very thin threads looking like nanotubes, and eventually they disconnect. So here are two examples, one connected, one disconnected. And they analyzed different types of neurons and showed that some of them do not make exophers and others make a lot more exophers than others. I don't remember, I'm not a C. elegans person, so I don't remember exactly what all of these acronyms stand for, but the ALMRs form by far the most number of exophers. How is it connected to protein aggregation? Well, the hypothesis is that this is taking out the trash. So when the cells might be overwhelmed by protein aggregation, they may resort to this mechanism, and they tested this hypothesis with several different proteins. One of them was Huntingtin exon 1 with a long stretch of glutamines. This is shown here, labeled in blue, and it's pushed into the exopher. On the background of GFP, similar results with M-cherry. Here is the exopher with a lot of M-cherry and the Huntingtin, and they showed that it doesn't happen to the same extent with normal Huntingtin with either no or short polyglutamine stretches. Similar results were observed with A-beta. When they expressed A-beta in the worms, they found that it was pushed into the exophers. And they actually took advantage of the fact that of the two fluorescent proteins they used, GFP and M-cherry, GFP remains soluble, but M-cherry tends to aggregate. So a lot of the subsequent experiments just used M-cherry itself. And they show here an example of a cell with an exopher. The GFP remains in the soma, whereas the M-cherry is pushed mainly into the exopher. And this is quantified in this graph here. They then asked, okay, so if we disrupt some of the cellular mechanisms that take care of degrading proteins or folding proteins correctly, can we increase the number of exophers? And the answer was yes. So they did that both with pharmacologic inhibition or with RNAi knockdown. They showed in particular that blocking the chaperone network by knocking down HSF1, a master regulator of chaperones, increased the number of exophers by about 6-fold. Blocking autophagy also increased exophers, blocking the proteasome increased exophers. So all of these data supported this idea that the exopher's job is to take out the trash. And this is a summary of several images, both fluorescence microscopy and electron microscopy, trying to look deeper into what's inside the exophers. So here's the soma of the cell with the normal organelles, and they could show that in the exophers, you can find pretty much every organelle they looked at: lysosomes, ER, some things they called empty vesicles, there were lysosomes, there were aggregates. So it looks like the exophers can take pretty much every component of the cell except the nucleus. And when they looked at where these exophers end up, they took advantage of the fact that the nematodes are transparent. They could see that the M-cherry aggregates in exophers first traveled to neighboring hypodermic cells, but those cells were not able to do much with the exophers. So then a few days later, the exophers ended up in cells called coalomocytes, which are macrophage-like cells. And there they did see degradation of most, not all, of the M-cherry aggregates. And they saw that over time, there was more and more of this fluorescence moving from the original neurons through the hypodermic cells to the coalomocytes for degradation. Some additional observations in that original paper were that they did see some exophers forming under native physiological conditions, so they didn't have to induce it by expressing aggregating proteins. The exophers were not limited, as I said in the beginning, to particular cell types. They saw different types of sensory neurons, dopaminergic neurons, producing exophers, just not as much as the ALMR. When they analyzed the ALMR, again, taking advantage of the fact that you can observe these things in living nematodes, they saw that if a neuron expressed or had an exopher early on in life, it actually responded better to light touch later in life, meaning that the exopher helped it maintain its health. So one of the hypotheses they formed was that it's not just a trash removal mechanism, but that it may serve also as a normal component of an orchestrated proteostasis reset in young adulthood, involving the removal of neuronal debris generated during development. And I don't have time to show you all of this, but there is a peak of exopher formation at a young age, then a trough, and then again an increase at an older age when the worms start to accumulate more aggregated proteins. They then followed up with a paper last year in which they tried to identify more closely the stressors that will cause exopher formation and the underlying mechanism. And a surprising result there was that the strongest inducer was fasting. That increased exopher formation by about 10-fold. Other stressors such as lower or higher temperature did not do much. A little T-jump increased by a little bit. Osmotic stress, oxidative stress did induce, but not as much as fasting. And I don't have time to go into mechanisms, but they did make some mechanistic observations there, suggesting that at least two different signaling pathways were involved, the EGF and FGF. If anyone is interested, the paper is out. They did show that if you increase the stress above a particular threshold, you actually get suppression of exopher formation. So apparently the cells can deal with it up to a certain point, and then not clear exactly, but they seem to give up. And that was shown both for osmotic stress with different osmolytes and with oxidative stress, with juglone, mitochondrial toxin, and with two of the famous Parkinson's toxins, rotenone and paraquat. You increase up to a certain point, and then there is a decrease. And also, if you combine stressors together, for example, fasting with some of those other stressors, you get a decrease. So the cells cannot deal with too much stress using exophers. They deal well up to a point, and then if you increase the stress, the exopher genesis shuts down. So with all of this information in mind, we asked some questions, and I'll show you how we came about these discoveries, but questions that came to mind were, are exophers unique to C. elegans, or are they found in other organisms? Obviously, mammals are interesting for us. Are they exclusive to neurons, or do other cells make them? And are there other roles for the exophers other than trash removal? So how we came about this discovery was by chance. We were working on analysis of a mouse model of tauopathy that was treated with one of the molecules that Raz mentioned in the introduction, CLR01. And the first postdoc who made this discovery was Jing Di, who later moved on to University of Kentucky, where she is a pathologist now. Jing observed this neuron in the mouse brain, big neuron full of phosphorylated tau stained with 88, that had this extension, a nanotube that ended up with a globular structure that she thought might be an exopher. She sent me the picture. We thought it was really interesting and started working on that. When Jing left the lab, Ibrar Siddiqui joined and took over the project, and he's now leading this project. He's in the audience, and I hope that he will write down all of your interesting questions at the end. So one thing we discovered in the beginning was that it was hard to find the exophers in mouse brains. They are a rare phenomenon. Unlike the worms, where you can grow hundreds of them, we didn't have this luxury. And when Jing started looking at many sections, she found very rare exophers. So we decided to switch to primary neurons in hope that it will be a little easier. And generated primary neurons from 3 types of mice: wild-type mice, the PS19 tauopathy model that has the P301S substitution associated with frontotemporal dementia, and wild-type, H-tau mice that has wild-type human tau, all 6 isoforms on a mouse tau knockout background. We did find more exophers in those cultures. One of the initial observations was that the size of the exophers varied very widely. There is over an order of magnitude variation from very small ones to exophers that are basically the size of a cell. And one of the common questions is, so how do you know that it's not a cell? Well, the answer is it doesn't contain a nucleus. So there are no current markers for exophers. You have to identify them morphologically, and you have to stain for nuclei to make sure that this is not a cell that you're looking at. But you see the examples here, connected via a nanotube, evaginating from the cell. Here it's hard to see, but this is also emerging from the cell. We then quantified these exophers, and we observed that there were many more in the tauopathy model. and that the number of exophers was fairly low in these two, in the wild type and this model in the beginning, but increased with the age of the cultures, doubled or tripled, and then remained stable between 2 weeks and 3 weeks. In this model, interestingly, there were a lot more exophers, and they were apparent already at day 7. We then asked in the tauopathy models, how do the exophers that contain tau, the tau plus exophers, compare to the tau minus exophers, and made some interesting observations there. So here are the exophers in the P301S mice. These are the H-tau. When we quantified them, you can see that in the P301S mice, in the beginning, there are a little more of the tau minus, but after another week in culture, there is a substantial increase in the tau plus as the exophers start to do their job in trash removal. For whatever reason, we don't really understand it. In the H-tau mice, in the beginning, in the young neurons, there are a lot of the tau minus exophers, some of the tau plus, and then it changes and we get a lot more as the neurons age and need to get rid of the tau. There's also a tendency in this experiment for the neurons that contain tau, so for the exophers that contain tau to be larger on average than the ones that do not, but this, as you can see in the numbers, is not always the case. We then wanted to do some more analysis and some more observations, and working with primary neurons is also not the easiest, so we decided to look for a simpler system and ended up using the SHSY5Y neuronal cell line. It's a little easier to find exophers there, and you can do more light microscopy, a little more easier. So Ibrar tells me that he can find an exopher in cells that are not manipulated, about 1 for every 30,000 cells. And he made some really interesting observations, such as exophers can be connected to more than one cell. So as shown in this image, there is an exopher, looks like it's traveling between 2 cells connected by a nanotube. This one is even connected to 3 different cells. There are also exophers that appear to kind of kiss or touch each other. And this all dovetails with the observation that even in the wild type young neurons, there are some exophers, suggesting that there is another role for the exophers that is not just trash removal. We don't really know what it is. We hypothesize that it's mediation of some short-range communication and transfer of cellular material between neighboring cells based on these images. The next question we asked was, can we see different organelles in the exophers the same way that the C. elegans researchers saw? And the answer was yes. Every organelle we looked at, including mitochondria, ER, autophagosomes, Golgi, and lysosomes, was found in the exophers. This is the SHSY5Y cells. We did not find, interestingly, every organelle in every exopher. They are about 80% of the time positive for each organelle we looked at, but then there is a 20% of the exophers that do not show the staining for each one of those, making things a little more complicated. We have started to look for stressors to see if they can increase exophers. These are new results that I'm showing here for the first time. So here is a lucky case in which in one particular field, Ibrar could see a couple of exophers in untreated cells, but when he treats them with sucrose at the optimal concentration he found of 250 millimolar, then the number increases quite dramatically, and that's quantified here. And it's also time dependent. It looks like 4 hours is the optimum, and by 8 hours it starts to decrease. We looked at whether HSF1 could increase the number of exophers, similar to what the people who did the study in the C. elegans reported. We were fortunate to have in the campus a mouse model that has that is heterozygous for an HSF1 knockout, so it has only half of the amount of normal HSF1. And surprisingly, we did not find the same finding as was reported in C. elegans. Neither the exopher frequency compared to wild type mice, nor the exopher number or area changed between wild type and the HSF1 heterozygous knockout mice. One of the interesting questions is whether exophers contain the phosphoserine flipping of the inner membrane to the outer membrane, which is an eat me signal for immune cells to come and digest and phagocytose the structure. That's very common with apoptotic cells. And we asked whether this is found in the neuronal exophers. The answer is no. And I'll show you in a couple of slides that this is different from some other types of exophers. So here you see the apoptotic cells are staining strongly with annexin 5 that binds to phosphatidylserine, but the exophers absolutely do not. Lastly, we asked whether exophers can be found in the human brain. So Ibar looked at several brains from controls, Alzheimer's disease, frontotemporal dementia, and PSP. And the answer was yes, he could find exophers in all of those. There were fewer exophers, and he needed to work really hard to find them in the control brain. You can see all types of exophers, those that evaginate directly, those that are connected via a nanotube, and those that have already disconnected. There were increased numbers in all of the diseased brains. And again, there was not really much of a change in the size of the exophers. The size range still varies widely. Of course, we wanted to know if the exophers in the diseased brains are affected and colocalized with tau and A beta. So this is summarized in this very busy image here. You can see the control brains here. This is just beta-3 tubulin to mark the cells in the exophers. There's no tau or A beta, and you can see the exophers that are free of anything. AD brain, you can see both A beta and tau. Interestingly, the exophers can colocalize, as is shown here. This big exopher colocalizes both with A beta and with some tau. This one does not. So they are not just a trash removal mechanism. Similarly here, FTLD brain, here's an exopher that does not colocalize with tau. This is a PSP brain, and here's an exopher that does colocalize strongly with tau. So there's still a lot to learn about it. It's a really nascent field, very interesting, and we're trying to understand what is going on. I didn't have time to tell you that there were a couple of studies reported in the last 2 years of exophers of other cells, specifically muscle cells. One study in C. elegans, one in mammals. In the mouse and human, it was mainly a mouse study, and it was specific to cardiomyocytes, not all muscle cells. The C. elegans was body muscle cells. So it based on these studies, it looked like what the authors of the cardiomyocyte paper called exophers. I'm not sure that they are really exophers because they differ by quite a few criteria. They are very small. very compact size distribution, and they do have the eat me signal. The C. elegans muscle exophers do look more like neuronal exophers, but one interesting observation is that unlike the neuronal exophers in C. elegans that are increased in response to blocking of autophagy, the C. elegans ones are, the muscle ones decrease when autophagy is blocked. And here is our data. So it's larger in size than what is in the C. elegans, as you might expect, because mammalian neurons are larger, but the same fraction of the cell volume. Overall, no eat me signal, no response to reduced chaperones, as I showed with the HSF1 heterozygous, and we don't know yet. We're doing the experiments now to see how autophagy affects the number of exophers. So just to summarize, this is a totally new field, started 5 years ago in C. elegans, structures that participate apparently in maintaining proteostasis, but also apparently do other things. They can be upregulated in response to multiple stimuli. We are the first group to discover them in the mammalian brain. The exophers, as I mentioned, contain various organelles, but never a nucleus, and this is the distinguishing factor that allows us to find them and distinguish them from cells. The exopher number, but not the size, increases in Alzheimer's and other tauopathies compared to the control. Primary neurons of tauopathy produce more exophers than wild-type neurons. And after a couple of weeks in culture, there are more abundant tau plus exophers than the tau minus exophers, supporting their role in garbage removal. The presence of the innate exopher, those exophers, those that are in control brain or young mice, suggest that they may mediate short distance communication, but we have yet to establish that. And as I said, some rare exophers were observed to be connected via nanotubes to more than one cell. So I just want to thank Ibrar and Jing, who have done all of the work, and my collaborators who donated the human brains and who helped us with statistics and the funding from NIH. I'm happy to take questions.
So,
thank you
am I supposed
very
to
much,
share the...
Gal. I'm very, oh, sorry.
Thank you, Gal. Very, very nice talk. I learned a lot on novel things. Just before moving to the Q&A session, if it is possible, I would just take a couple of minutes to remind to the audience that we have to advertise an important upcoming event. The International Symposium on Pathomechanism of Amyloid Disease, the first in person of the post-COVID era, will be held in Catania, Sicily on August from 25-27. This 3-day symposium will host about 30 invited speakers who are experts on amyloid-related research. There are poster presentation and a lot of opportunities for networking and discussion, students' participation, and last but not least, an excursion to some exciting sites in Sicily. The total number of participants will be restricted to a maximum of 100. So registrations will be accepted strictly on a first-come, first-served basis. And this implies that if you are interested to participate, you should not hesitate too much to register. I have cut and pasted the flyer in the chat box, so the audience could download from there. Now we can move to the Q&A session. I can see, okay, from Martin Mushel, there are two questions. I'll read it. The first, any insights? What's the role of the long nanotubes connecting exophys to cells? And the second, if exophys are trash removal units, where are they dumping, dissociating? Where are they dumping, dissociating the proteins they contain?
Okay, thank you, Martin, for these questions. So what we have seen and some other groups have seen is a really remarkable connection between the cell and the exopher as long as they are still connected via the nanotube. You can see small packages sent from the cell to the exopher as if the cell formed the exopher, put all the trash there, but then, found some more and is continuing to send that into the exopher until it decides, okay, now it's time to disconnect and let someone else deal with this pile of garbage. Now, the hypothesis is that the cells that will deal with the pile of garbage obviously are glia, microglia, astrocytes possibly. We have not yet shown that. We're doing these experiments right now. In the first image I showed you, it looked like microglia were touching the exopher, but that's one image. So we're working now on trying to establish whether or not the material in the exophers ends up inside those cells. Those experiments are in progress. So I think that also addresses the second question, but there is another interesting aspect here. If that mechanism is not working properly, then the exophers might be one of the mechanisms, maybe a major mechanism, by which proteins such as tau, A-beta, synuclein may spread. If the exophers do dump their contents and it's not all degraded, obviously that could contribute to the spreading. Danilo, do you want to read the second question, or I can read it?
No, if you want, I can read it. From Maru Jason Velayutan, and sorry for the horrible spelling, but the important thing is the question. What about interaction between exophys and neutrophils? Right,
so we have only looked at exophers in neurons or in brain. As I said, we have not yet established where the material goes, but I think that you would have to see neutrophils entering the brain in order to address this question, or to study other types of cells if they form exophers, and then they could interact with neutrophils in the periphery.
Okay, we can move to next question from Yan Zhong Wang. Few question: does exophy formation require cytoskeleton? If so, which one? The second, when you have a mixture of soluble and aggregates of tau or other neurotoxic proteins, are they selectively enriched in exophys? And third, I assume the brain samples are 10 micromolar, no, 10 micron cryosection. If so, how do you know the exophy you detect are not corners from other neurons? Please.
Okay. Thank you for all of these questions. These are all great questions. So does exopher formation require a cytoskeleton? The answer is obviously yes. There must be cytoskeleton proteins that support the nanotube and the exopher itself. We have not looked at all of them exhaustively, but we looked at tubulin and actin. We found both of them. And again, one of the confusing observations is that you don't always find the same thing. So in some you see a lot more actin, in others you see a lot more tubulin. The staining can be stronger in the exopher than the cell, but not always the case. So it's really complex, and we're only beginning to explore what exactly is happening there. When you have a mixture of soluble and aggregates, are they selectively enriched? Great question, we don't know yet. That's under investigation. And how do we know that these are not just corner of other neurons? Again, what I said is we identify the exophers at this point only morphologically. So in some cases, when they appear to be completely disconnected, they could be corners of other neurons. So we try to focus really on the cases in which we see the connection, either evaginating directly from the cell membrane or still connected via the nanotube.
Okay.
Great talk, Gal. I really enjoyed it very much. How different are these exophores from exosomes, which is also play a big role in the intracellular communication? So I wonder these exophores can play a role in fueling protein aggregation also.
Mm-hmm. So exophers are much larger, and they do evaginate directly from the cell membrane, more akin to ectosomes than to exosomes. Exosomes, as you know, are formed in the endosomal pathway in multivesicular body, and that then merges with the membrane and releases the exosomes into the extracellular milieu. And there are different markers in exosomes that were not found to be enriched. But in the C. elegans people did address this question in their first paper and show that, those are two really distinct structures. I can tell you also that in one experiment, Ibrar looked at whether or not he could find exosome staining in the exopher. And the answer was yes, there was some staining of puncta that looked like exosomes inside the exopher. So two different structures.
Danilo, Binzu is on the panel. Binzu, go ahead.
Thank you, Rams. Dr. Bitan, very exciting talk, new direction. So I have two questions. So how do you quantify exophers? Do you have like a specific marker which is staining the nucleus and the tubulin, just the skeleton, so you can differentiate between exopher versus the neuronal body? But is there any other specific molecular marker mark the exopher? Right. The second question I have, okay.
So as I mentioned, the answer is no. We wish there was. There are no specific markers for exophers. So Ibrar spends many, many hours, and now we have a number of undergrads that help with that, spending a lot of time in front of the microscope, looking for these structures, defining them morphologically, and always looking for nuclear staining. So anytime you find nuclear staining, that's not an exopher. And as I mentioned, these are rare structures. So they're really poring over those microscopes and looking field by field until they can identify them. And then they obviously, count them and measure the size, and that's how they do the analysis.
The second question I have is you're showing some data in one of the slides showing human topsy like AD or PSP. They have a significant higher exopher area than controls. Those are the
area. The area did not change much because there is a large variability of the area in all the cases. In human brain, mouse brain, neurons, we always see a large variability of the size. What we do see is that in the tauopathy cases, there is an increase in number, significant increase.
Do you use the postmodern brain tissue, or I just wondering, how do you, those,
yes, these were postmortem.
Postmodern, but it is like they still, you can still observe those exopher there. I mean, is it after a certain period of time they will cannot be observed anymore, or?
These are all frozen sections. I can share this again and go to that slide. So here's the slide. As you can see, this is what we see when we stain for beta-3 tubulin and DAPI. This appears to be the neuron, and here's an exopher neuron with exopher. And then this, could be a disconnected exopher, or as someone suggested, could be just a fraction of a neuron that does not contain the nucleus.
Thank you, very interesting.
Shall I read the other questions?
Maybe Avi has a question, can ask. Right
Yeah,
now.
if I may. So really enjoyed your presentation. That was really nice work. So I was wondering, how do you differentiate between an exopher and an exosome? Is there any difference between the two?
Right. So yes, the answer is yes. As I told Rams, this question was addressed in more detail by the people who did the first study in the C. elegans, but they really are very different in size. Exosomes are much smaller than
yeah, but you have
exophers.
microvesicles that are of different sizes, So
Sure. Right. So the ones that evaginate directly from the membrane are called ectosomes, and they are still quite a lot smaller. They can reach a size that is near that of exophers, but they are not connected via a nanotube. They just evaginate and are released into the extracellular milieu. There's also a phenomenon called blebbing, but it's much shorter lived of things evaginating from the plasma membrane. The exophers, I didn't show the data, but they can sit there for hours and they can expand, they can contract a little bit while they are still connected via the nanotube, and the cells continue to send material into the exophers. We can see little packages going into the exophers until at some point the cell decides, okay, now it's time to disconnect.
Interesting.
And I also mentioned that we looked for exosomal staining, and we could see puncta stained for exosome staining inside the exopher. Okay, so the exopher is much larger, and it can contain exosomes sometimes. It basically can contain any organelle we have looked at, except for nuclei.
What about viruses? Can they be incorporated into an exopher?
I'm sorry, I didn't hear.
What about viruses? Can viruses be encased in here?
That's a great question. I don't know. Maybe we'll start a collaboration.
Because typically we think about viruses just budding off the cell membrane,
Right.
But maybe if they're encased in yet another membrane, that could protect it from immune attack, if that's the case.
That's a very interesting question. We have not looked at any infected, or tissue that we know is infected with viruses. But that will be really interesting to do.
I have a question from myself.
Yes.
You mentioned that proteostasis equilibrium disruption stimulates exopher generation. But I wonder, what about exogenous addition of amyloid? Is this able to induce the formation of exophers?
We're doing these experiments. I don't have the answer for you yet, but it's a great question, and we're looking at that.
Okay, so keep me updated about that.
Okay.
Okay.
Will do.
We will move to, I will read the other question from the QA box. The next one is, thank you for the presentation. In cell culture, do exophys always stay attached to the substrate, or can they also be released into the conditioned media?
They can be. So if you just grow them in cell culture, you know, we are always concerned about misidentifying them. So when we just see something that has, you know, looks like a small sphere in the cell culture, we're concerned that this is just cell debris. So when we do the cell culture experiments, we tend to focus more on those that are still connected by a nanotube. Now, sometimes we do the live imaging experiments, and we take images over many hours, so you can see the exophers disconnecting eventually, and then you know this is an exopher. But without a specific marker, it will be really hard to isolate them and to say, okay, what I'm looking at here is an exopher that detached, rather than whatever other vesicle or little piece of debris.
Okay, let's move to another question from the QA box, from again Yanzong Wang, following Ram's question. How different are they from migrosomes?
I have not specifically looked at migrosomes, so I cannot answer this question. We will have to take a look at that specifically.
Okay, next question. Dr. Bitan, do the organelles in exophys function? And what is the size of the protein aggregates in exophys?
It appears to be similar to protein aggregates in other places where you look at them. So when we stain them, I mean, I can show the screen again if people are interested. There is no big difference between the size of the aggregates that we see in the exophers and the size of the aggregates that we see inside the cells themselves.
Okay, we have another question from Simon Heigal. Thank you for the nice talk. Do you already know anything about the biogenesis of exophys? Are there differences between different cell types and or organisms? Second, how can you differ detached exophys from larger extracellular vesicles such as microvesicles?
Right. Let me see. So anything about the biogenesis at this point, what we know is what the C. elegans people have told us. In their second paper, they have looked at the biogenesis. They showed that especially under the fasting conditions, there are a number of factors participating in the exopherogenesis signals. I mentioned that both the EGF and the FGF signals participate. There are also, if I remember correctly, metabolites of well, no, I don't remember correctly. So I'm sorry, but I will need to refresh my memory about exactly what is involved there. What they did show quite convincingly is that it's not necessarily a cell autonomous phenomenon in the C. elegans, that the whole organism participates in the sensation of the food deprivation and the then formation of exophers in neurons. The second question was differentiate.
What about, yes.
So microvesicles, to my knowledge, do not extend beyond, I would say, 1 to 2 micrometers. Exophers can be much, much larger. But the main thing, as I mentioned, is once they detach, The small exophers you cannot really differentiate because there is no specific marker. So we say that something is an exopher when it's this small, when it's still connected via the nanotube. Then we call it an exopher. Otherwise, we don't know.
Okay, we have a last question from the box from Marujesan Velayutam. What about HIV-Tat protein complex formation in T cells? I guess this is for the first speaker.
Yeah, so you're absolutely right. We haven't studied it in great detail, but our experiments do suggest that formation, complex formation in T cells as well. The only problem with T cells is that when HIV replicates in T cells, it's cytopathic. So the T cells will just burst and then don't stay intact. And so that's really the problem. Well, you know, macrophages and astrocytes, they will stay intact. You don't see cytopathic effects on them. So there it's a lot easier to study it.
Well, I don't see any further question, but not sure that I don't see any further question. No. No, there is a hand rising. I don't know.
I just promoted her to the panel, so we can.
Yeah.
So meanwhile, so why can't these exosomes or exophors, which are communicating between cells, can they play a role in transmitting the infection and enhancing the infection rate by bacteria or by even the amyloid aggregation itself? Can one cell transmit toxic species to another?
Right, that's exactly what we think. I mean, I don't know about bacteria. You know, we're talking about much larger structures here, but the amyloid aggregates or viruses definitely could be transmitted via exophers. We are working now on testing whether it happens with amyloid proteins. We don't have the data yet. And as I told Avi, I have not done any experiments with viruses at this point, but could be done.
Beena, your turn. Beena Krishnan, yeah.
Thanks, Ams. Sorry, it would have taken longer to type out the question. So have you looked at, this looks like another mechanism for protein degradation. So the cells that tend to form exophers, do they have reduction in other degradation machinery? For example, autophagy is to a lesser extent, or proteosomal degradation is to a lesser extent compared to, and therefore this happens to be a key mechanism to get rid of the aggregates.
Right, so this is the hypothesis that this is an answer, and a newly discovered mechanism that answers what the cells do when there is too much of an increase in proteostatic stress and the regular degradation mechanisms don't work or cannot manage all the proteostatic stress. In the C. elegans studies, they have shown that if you decrease autophagy or decrease proteosomal function, you get more exophers. We are still doing these experiments. We could show that with osmotic stress, we can increase exopher number. And I mentioned that one of the interesting observations is that in muscle cell exophers that were also discovered in C. elegans, decrease in autophagy actually decreases exopher formation. So it looks like the muscle cells use this mechanism in a different way, and it may be actually autophagosomes that are released out of the cells. That's the way it looks to me just from reading the papers. Whereas in neurons, the exopher formation is a different mechanism that is increased when you have decreased autophagy.
I see that if you reduce the other degradation machinery and you see an increase in the exopher formation, but under a natural condition when you are staining the cells to see the or you are looking at the fluorescent aggregated proteins. If the same cells, if they are stained for markers for, let's say, autophagosomes or any proteosomal markers, then in the same cell, you see that there is indeed a reduction in the other machinery and there is an increase in the exopher formation under a natural scenario.
Right, so those are all great questions. The limitation here is how rare those structures are. So under normal conditions, it will take some time to do these experiments, to stain on the cells and then find those that contain exophers and try to quantify whether there is a change in the cells that have an exopher in the staining for autophagy or proteasome compared to all the other cells that do not. We may try to do this under the conditions that increase exopherogenesis, but that's already a manipulation, and we don't know exactly how the manipulation, for example, like osmotic stress, affects proteasomal degradation and autophagy. So, good question, difficult to answer.
Yeah, I think so. Thank you.